Hello and welcome to a quick overview of the final concept in Chapter 5, which is cell signaling. Now, in multicellular organisms, so think humans, giraffes, trees, there needs to be a mechanism for cells in different types of tissues to respond to each other. And that primarily has to do with the fact that as multicellular organisms became more and more complex, they evolved different types of tissues that specialized in different types of systems and roles. So your stomach cell is primarily evolved to digest protein, but it needs to have signals from other molecules and from other cells in order to know how to do its job appropriately. And the communication between these cells is really also important for many unicellular organisms on a different scale, and that usually has to do with population dynamics and also thinking about resource utilization. So there's three different types of signalings that are important to understand their roles. So you can have paracrine signaling, which is very short distance local signaling, where generally there are going to be secretory cells that are going to be releasing specific signal molecules to target cells nearby. And usually that's going to be resulting from vesicles being fused with the membrane and those molecules being released into maybe an extracellular fluid or the bloodstream, something that's going to result in the cells being targeted. Another type of signaling is synaptic signaling, and this really occurs with neurotransmitters, neurons specifically, and target cells. So we have the synaptic cleft between the synapse of a neuron and also another targeted cell, and that's really going to be, again, short distance signaling. It's just that there's an electrical signal that causes the nerve cell to release the neurons rather than probably a protein being expressed within the nucleus and paracrine signaling. And then we can have long distance endocrine signaling, where we are either going to be using hormones that travel in the bloodstream or with a chaperone molecule that can go into the cells or bind to the receptor and cause cells and tissues far from the source cell to elicit a response. And signaling was really discovered recently by scientists based on just examining organismal cause and effect. So, for example, when your body is under stress, you see a bear, you're anxious about school, you often enter what is known as fight or flight. And that has one possible outcome where your kidneys might produce epinephrine. And epinephrine then causes a lot of things to transpire within different organs, such as muscle cells or your lungs or your heart. And there's three main processes that have the source and the response occurring. One is reception, two is transduction, and three is response. So the reception is always going to be a signal molecule binding to some receptor protein, either in the membrane or in the cytoplasm, which is going to cause transduction, which is really relay molecules or intermediary molecules being activated or turned on within the cell. And then the response will be some cellular change. Often it is protein production or cellular division or a complete new round of cell signaling to other cells. So the most important thing to know about reception is that it is the binding of a signal molecule to the receptor protein, and there's always going to be a special structure between those two molecules that allows for a specific reaction to occur. So one signal molecule is only going to be able to target one specific type of receptor. So we keep those messages discrete and specific, and when those bonds form, it is called a ligand, which generally causes some conformational shape within that protein. If you remember, proteins have a very specific tertiary and quaternary structure based on bonds between amino acids. So if we have another protein form and bind to that receptor protein, then that's probably going to quote unquote activate them. Many water-soluble signal molecules, because of the hydrophilic nature of the plasma membrane, have to bind on specific sites in the receptor proteins. And there's two types of proteins that are important to just understand and know the differences. One is the G protein coupled receptor, and the other is the ligand-gated ion channel. So the GPCR, or the G protein coupled receptors, are plasma membranes that work with the help of a G protein. G proteins bind to and use energy-rich molecules of GTP, which are basically a cousin of ATP. And the G protein basically acts as an on-off switch. If G protein is found next to or bound to the G protein is inactive. And if the G protein is activated, then GTP will be used. Many G proteins are similar in structure, showing that there's some evolutionary novelty within them. Many organisms share the G protein, but they're very diverse in their function. So the structure is similar, but their application is very different. So right now we have a activated GPCR molecule, and we have GTP bound to the activated G protein. So we have right now the first part, we have reception. A signal molecule that's water-based has been bound to the activated GPCR, and we have a G protein that's embedded in the inner part of the membrane that's going to then cause the activated enzyme in the membrane to be turned on. And notice when we have this G protein leave the receptor, the ligand is detached. So this is a way to make sure that a signal is discrete and does not continue forever. We're basically inactivating the reception. Now, alternatively, a ligand-gated ion channel acts as a gate for ions to come in and out. When a signal molecule binds as a ligand to the receptor, it allows ions such as sodium or calcium through a channel in the receptor. The reason why these molecules need a receptor is that they're charged, so they're not going to be bound to the plasma membrane easily. Ligand-gated ion channels are very important in the nervous system in terms of resetting membranes to have a unidirectional specific messaging system. If this didn't occur, your nerves wouldn't be able to reset, so touching hot water or feeling the burning sensation on your fingers if you chop some jalapeno peppers wouldn't really change, and that could be a really bad thing for you as an individual. So what happens often is that we have some preset concentration of charged membrane molecules. So here are some ions outside of the membrane, and a signal molecule is then going to bind to the receptor protein, which is going to allow these molecules to flow from high to low. So we have some diffusion occurring. Again, this is facilitated diffusion, so we need a membrane-based protein to let the molecules in. And this flooding of ions is going to change the inner biochemistry of the cell, which is in turn going to elicit a cellular response. Again, these signals are fleeting and discrete. Discrete meaning that they happen once and then it resets. So if there's another signal, it can occur again. So the gate is closed when the ligand is broken apart. Now, some molecules, hormones specifically, that are lipid-based, they're formed off of cholesterol, hello, chapter 3, are not the same as all of our water-based molecules. And the reason why has to do with basic properties of the cell membrane. Hormones like testosterone are lipid-based, so they can easily diffuse through the plasma membrane and enter the cytoplasm. And when they do that, they are met with a receptor protein, or basically a chaperone, that basically forms a ligand, so a really specific protein with a very specific opening for that hormone to bind to. And that is almost always going to result in a cellular response that we would call the expression of a protein. So secondary sex hormones like testosterone and estrogen are really important in having specific types of cellular responses throughout tissues in the body that are very dependent upon age and sex and also concentration of those hormones. So men and women both have testosterone and estrogen, the difference being how those levels are expressed on the daily or monthly value, but also how those then target different tissues in the body throughout puberty. So the second big component of molecular signaling is transduction. And this is the more confusing, chaotic aspect of cellular signaling. I sort of think about cellular response, or the first part of cell signaling, as someone screaming fire in a big building. And then signal transduction is what happens inside the cell after someone screams fire. What are all of the different processes and pathways doing in order to respond to that one stimulus? There does exist two different things that are important to know. There are multi-step pathways that can amplify a signal, meaning a few molecules of a reception can lead to a huge cellular response. But also the multi-step pathways provide more opportunities for regulation of cellular response. So multiple responses can occur from just one signaling. It'd be sort of like me screaming fire right now in the library. I'm looking at it right now as I record this, and some of the students might be trying to put the fire out. Some of the students might be trying to organize to go outside and be safe. Ideally, we'll all leave if there's a fire, but this is just honing in on the point that there would be a coordinated, complex response to it. Now, phosphorylation and dephosphorylation are the mechanisms for regulating the proteins that are involved with transduction. Phosphorylation, if you remember, is part of the ATP molecule, and phosphates are very charged negatively. So phosphorylation, in terms of ATP and ADP, are going to either release or expand upon a lot of energy. So if we use ATP to add energy to a protein, it's going to activate it, and then when the ADP is taken away, it will be inactivated. So often what happens with phosphorylation cascades is we have a signal molecule, it binds to a receptor, a ligand occurs, and then we might have a G protein or an enzyme that then causes the first protein to be formed. And this protein is going to go from inactive to being active, which means it's going to use some ATP energy to go activate a second protein kinase. And then this protein will be inactivated after it does its job to activate a protein by having that phosphate group removed, and it will be recycled back into being inactive. So every time ATP is invested, we're activating an enzyme, and then that enzyme or that protein kinase will then be inactivated when it loses those phosphate groups. So protein phosphatases, the PPs, are a really specific type of protein that removes those phosphates from those proteins. It's basically a regulatory mechanism. They're basically the teacher or the fire marshal saying the fire is over, you can stop being really worried about being in the school. There's no problem here. And this is really important for making sure that we can reuse the molecules for a second or third signal, but also that the signal has a discrete response in terms of time and also action. Now, the extracellular signal molecules, the ligands that bind to the receptor, is the first messenger, but there are secondary messengers that are in the cytoplasm that can cause the cell signal to be expanded and amplified. And one of those is cyclic AMP and calcium ions that are used to sort of activate more and more responses. Cyclic AMP is one of the most widely used messengers. And what happens is adenyl cyclase, ASE, an enzyme, in the plasma membrane rapidly converts ATP from cellular respiration to CAMP in response to different types of signals. The immediate effect of CMP is usually the activation of protein kinase A, which then phosphorylates a variety of other proteins. So CAMP is really a way to get the primary messenger, the first messenger, amped up, so to speak, amp up the message to be more distributed. So what often happens is we have our primary messenger with GTP being activated. GTP, once it's removed from the G protein, will inactivate this enzyme. But adenyl cyclase is going to be the enzyme that takes a bunch of ATP and converts it into CAMP, which will then go and create protein kinase to be activated. So either by secondary messengers or by using ATP, we can create diverse and also amplified responses. Ultimately, a signal transduction leads to a cell activity or cell response. It might be in the cytoplasm. It might be that the organelles start to replicate and separate for mitosis. It might be the synthesis or regulation of an enzyme or a protein by turning genes on or off. The final activated molecule in the signaling pathway also might function as a transcription factor, which we saw with the hormone testosterone, but also with a water-based receptor, we can have a gene response as well, where we're going to be making some sort of protein. This could be your pancreatic cells creating insulin, which is a protein-based hormone, as a result of your body having high blood sugar. So, in a nutshell, when you have a signal molecule released from some specialized tissue or some change in your body, there's going to be a reception, which leads to some level of transduction using secondary signaling and ATP to activate molecules. That eventually will lead to some transcription factor going into the cell, into the nucleus, and responding with some protein production or gene expression.